Alright everybody, welcome to lecture 11. I was actually going to skip this and go eat food and then come back and do it, and then I realized that a lot of the slides are just repeating of lecture 10, so I'm actually going to go ahead and take a look at this. Yeah, let's go ahead and get to it. Lots of office hours, yeah. She did a quick clarification on hedgehog signaling. I like this image a lot more. This image is significantly better or significantly easier to understand. All you need to know about this is when the pathway is off, patch inhibits smoothened, and this glee factor just freely goes through, gets processed into a repressor, and it turns off these genes. When patched is turned off, when hedgehog is bound, smoothened actually is active, and causes the cleavage to not occur, and so it actually acts as an activator instead. So you can, like, when hedgehog is active, it leads to a lot of division, and then, or when hedgehog's present, and then when hedgehog is not present, it leads to how do you say it? It leads to a lot of defects where you have less limbs. So hedgehog present, more limbs. Hedgehog not present, less limbs. So yeah, it's like basically the patched inhibits smoothened. The glee is phosphorylated, and the phosphorylation leads to cleavage, turns into a repressor. When patched is inactive after hedgehog is bound, smoothened stops glee from being phosphorylated, so glee is no longer cleaved, and so it just acts as an activator instead. That's all I wanted to get across from that. Now, things to talk about in terms of what we already have spoken about. Already mentioned this in the previous lecture. Take a look at this one if you want. You take a look at my explanation, or Professor Kong's if you need extra opinions on how this works. And again, always default to Professor Kong's explanations over mine, because I'm a student. I get things wrong, believe it or not. So, yeah. Already talked about dynamic actin, and already talked about how movement is important, or how it's important in movement. Just remember that the plus end and the minus end are both capable of growing and shrinking, but the plus end grows and shrinks quickly, while the minus end grows and shrinks fast. This is very different from microtubules. They really only grow and shrink from the plus end. Really actually a cool video about how a neutrophil chases bacteria. So I actually found this pretty funny when I first watched it. So real quick, talking about actin, what are the subunits for actin? It's going to be the actin monomers, and it specifically uses ATP and ADP. And what motor proteins interact with actin? It's going to be myosin. And so we actually should be able to do this for microtubules. So what are the subunits for microtubule? It's going to be a heterodimer of alpha-beta tubulin. The nucleotide is going to be GTP/GDP. And the motor proteins that are involved with microtubules, specifically that you guys have talked about, are going to be dynein and kinesin, once we have talked about them a little bit more. So, yeah. Now, let's get into this. We're going to talk about microtubules today. We've already talked a little bit about them, so I'm not going to spend too much time. But remember that, microtubules have a plus and a minus end. The minus end of microtubules basically is at the nucleus, and the plus end of the microtubules is basically at the cell membrane / cell wall, whatever you want in terms of the cell you're looking at, whether it's plant or animal. So we already talked about a closer look at microtubules. The individual quote-unquote monomer for a microtubule, or the subunit for a microtubule is actually the better way of rephrasing that, is made up of two separate proteins, the alpha-tubulin and beta-tubulin. And these alpha-beta-tubulin subunits basically stack together to form a line of these this polymer, which is a protofilament, and then you stick these protofilaments in a circle to form a microtubule. The plus end, remember, is the one that has the beta tubulin facing it, and the minus end is the one that has the alpha tubulin facing it. And like I said, you circularize these things like this to make a microtubule. I'm actually going to switch to red because now we're looking at green stuff. So, I already talked about this stuff. Just remember, right, that the GTP binds to, specifically in our case, mostly it binds to the beta subunit, or the beta tubulin. In the GTP bound form, it has a high affinity for other tubulin molecules, and so it causes polymerization. When it's in the GDP form, so when the GTP is hydrolyzed, it has a low affinity for other tubulin molecules and tends to depolymerize. So when GDP is present, depolymerize, and when GTP is present, polymerize. All right. So, an interesting thing about microtubule dynamics is that it's actually kind of linear. So basically, the more free tubulin you have, the more elongation occurs. So the higher the elongation rate is. And so this point here, where it's 0, is basically this point of steady state equilibrium, where the free tubulin concentration is just enough where the depolymerization basically exactly counteracts the polymerization, if you will. So you can see here, when there's no free tubulin, there's pretty much no growth. Actually, it's depolymerizing. That's why it's negative, right? And then as you add more and more free tubulin, the elongation rate grows, and so it doesn't depolymerize as much in this section. Eventually, we get to this 0 point where the free tubulin amount exactly counteracts the depolymerization. And then as you add more of it, we get to this point where the elongation rate starts to spike drastically. So again, here is the shrinking state, so they highlighted it here. So this right here is the growing state. Remember, when we have more than enough free tubulin, we enter the growing state where the elongation rate far surpasses the shrinking rate. And here we have the shrinking state where we have very little free tubulin. The elongation rate doesn't counteract the shrinking rate, so the shrinking rate actually overpowers it. It's like adding two vectors together, right? So we have a growing vector and a shrinking vector. In this case right here, the free tubulin concentration is high enough to where the growing vector far surpasses the shrinking vector. So if you were to add these two together, plus this super small thing, you would get something like this, right? But in this case over here in the shrinking state, you have a super large downward vector, into a super large shrinking vector, and a small growing vector, and so you would get a large shrinking vector, if you will. It's one way to think about it. And so this put together is how you get this image here. Already talked about this. Remember, when GTP is on, it causes polymerization. When the GTP is lost, it causes catastrophe, which is shrinking. And then rescue is the process of gaining that GTP cap again, which causes the polymerization again. So, I know these answers are already circled here, but let's think about these logically and try and go through them one by one. So, if microtubules is growing with GTP capped end, is the free concentration of tubulin high or low? Well, if the growth rate was 0, then that would mean that, the amount of tubulin available is just enough so it cancels out the depolymerization. But since the tubulin is still growing at the GTP capped end, it must mean that we have a large amount of GTP tubulin, or a large concentration of tubulin, because, again, it's still growing. For this question here, if GTP hydrolysis proceeds more rapidly than subunit addition. Okay, so there's more hydrolysis, which means that it's more likely to dissociate, and it dissociates faster than how fast subunits add. So basically the cap is lost and it begins to shrink. Is the free tubulin concentration high or low? Well, it's going to be low in this case because remember, the rate of depolymerization is faster than the rate of polymerization in this case. So because of that, we can say that we were probably at a low concentration of tubulin. So if a new cap is formed and GTP-containing subunits resume adding to the shrinking end, is the free tubulin concentration high or low? So at this point, because we are back to growing again, the tubulin concentration has to be high. So again, the way you can think about this is like, when a tubulin shrinks, it has to release its tubulin subunits into the area. And so eventually it gets to the point where the concentration of tubulin is super high. And so it kind of goes back and forth, is a way of thinking about it. All right, now for motor proteins. Myosin, kinesin, and dynein. So the way that we can kind of think about this is these are all mostly plus directed. So, well, sorry, I should specify. So kinesin is plus ended. So the way that I actually remember this, right? So microtubules, right? They have their minus end at the nucleus and their plus end at the cell membrane, right? So kinesin and dynein are these walking proteins, remember? And myosin, we've already talked about, is the contractile protein. So kinesin is the is one of the walking proteins, and dynein is as well. So kinesin starts with a K, and kinesin is plus end directed. The way I remember this is K plus. And you should know this because lysine, right? I think about it as lysine plus, kind of, in a way. So lysine, I know, has a positive charge. So therefore, kinesin, and kinesin starts with K. So kinesin walks towards the plus end. On a similar note for dynein, dynein is D minus, and D, remember, is aspartate. So the way I remember this is that aspartate is negatively charged, and so dynein starts with the D, and the letter for aspartate is D, and dynein moves towards the minus end. So kinesin, right, the plus end, kinesin moves towards the cell membrane, while dynein takes things back towards the nucleus. Myosin is a bit different. Myosin, remember, from what you saw, is first of all actin based, not microtubule based. So kinesin and dynein are both microtubule based, so myosin is actin based, but myosin also moves towards the plus end. Remember how it cocks its head and it pulls? It is basically pulling itself to try and get closer to the plus end, is how you should think about it. And here's the video that you guys probably saw on the first day of class. Really funny looking video. But these guys put in work, man. 350 miles per hour ain't no joke. That thing is fast. Again, already talked about this. So dynein moves towards the negative end and kinesin moves towards the plus end, and they are involved in vesicle transport. It does say that some kinesins are minus end directed instead, but honestly, that's maybe something you put on your note card and forget about it. So, yeah. I've already talked about how kinesin and dynein move in terms of their motor proteins, so I'm not gonna, or in terms of their mechanisms, I'm not gonna harp on that. If you want to look at how I talked about that, check out the last lecture, but I'll very briefly say that ATP hydrolysis causes movement, ATP then replaces ADP here, hydrolyzes, causes movement, and so basically it's like a flip-flop back and forth where one leg moves out and then the other leg and one leg again, so right, it's like flip-flopping. So, yeah. I think this is probably actually a better showcase of how the mechanism works, because it's actually very similar to myosin and how it works. So again, just kind of going through each slide individually very briefly. I know I said I wasn't going to explain it, but I think these slides do a good job of kind of showcasing the intricacies of it. So ATP is bound, hydrolysis, we get ADP-PI. the PI is then released, and when the PI is released, it causes the movement. And while that movement is happening, the ADP on the other leg is replaced with an ATP. And so now this process repeats again. This ATP is hydrolyzed, the PI leaves, and then this shifts over, and this ADP is replaced with another ATP. And here is just a cool little video of how these things work. In vitro assays are cool little experimental techniques. And then here we can see a cool little thing, a cool little fact about them. There's actually 40 different kinesins and 16 dyneins, so just a fun little fun fact about them. Yeah, so these stories, I think, are important in principle to understand. So I'll go ahead and talk about them a little bit, but I won't harp on them too much. So microtubules, they're very important in development. So motile cilia and primary cilia. So motile cilia are like cilia helped in movement, and primary cilia, I actually can't remember exactly how primary cilia are super helpful. You guys talked about it in terms of hedgehog and stuff like that, so that's one way to think about it. But microtubules are important because, you know, without microtubules, you can't even form these cilia in the first place. Because here we have the plasma membrane is like this line here, basically going up. That's a horrible line, but you get the idea. And so basically, the microtubules are growing up this way. And so you can see here, these motor proteins are using these microtubules to travel up and down the cilia. So basically the cilia is able to gather resources and things like that from the central areas of the cell because the microtubules are able to move. And so the microtubules not only provide structure to make this kind of bulbous shape, but they also allow for motor proteins to travel back and forth so that resources and different nutrients and things can be shuttled into the cilia for whatever the cilia needs. So very important in that process. And here actually is a great example. You guys already talked about this in terms of hedgehog, but you know, GLI-FL doesn't just float around, right? It's carried through these motor proteins, through these processes. So it binds to these microtubules, it moves up, and then through maybe, so kinesin, it'll maybe move up, and then through a dynein, maybe it comes back down, and then it gets into the nucleus and does its activities. So a great little example of what these motor proteins kind of do and how microtubules are helpful in that, right? The cilia does not form without the microtubules, and the microtubule helps this GLI protein interact with patch, get degraded into a repressor, or helps it stay in the active form as an activator. So ciliopathies, or ciliopathies, I keep messing that up, ciliopathies, these occur, you know, when the cilia don't develop properly. So if they don't develop properly, the microtubules probably don't work, or some other motor protein, or the motor proteins that transport the cargo from the cilia don't function correctly. So there's plenty of places where you can get these. I think the intestines are very, intestines involve a lot of microvilli and cilia, so the intestines are a great place where this tends to happen, I believe. So yeah. And the second one, we already talked about this very briefly, is cell division and cancer treatments. So microtubules are incredibly important in cell division because here are the microtubules right here. They come out of the centrosomes, right? They help with organizing these chromosomes in these lines. They help with pulling them. They help with all of that. And actually the cleavage furrow here, like the division here, is actually caused by actin and myosin. Fun fact, actually. So dividing cells, they need dynamic microtubules. They need them. They're very important because microtubules take part in prophase, and prometaphase, and metaphase, and anaphase, just everywhere. They're super involved. And I like how this is here. This is like they haven't deleted their cursor before they took the screenshot of it or something. But anyways, you can imagine where, you know, if you use Taxol to prevent depolymerization or something, the microtubules can't be dynamic. And then on a similar note, if you use vidcristin or nicotazole to cause microtubule depolymerization, then the microtubules won't polymerize when they're supposed to. And so you actually just end up messing up this whole process. And so this is great for anti-cancer drugs because remember, at a certain point, the cells in your body will stop dividing. They don't divide anymore, at least most of them. But cancer cells always divide. They will continuously divide over and over and over again. And so these drugs, have a massive effect on cancer cells because those cancer cells are trying to divide and you're basically stopping them from doing that. And so the cancer cells will try to divide, they'll produce deformant cells, and those cells will just get killed. The last type of fiber we're going to talk about is intermediate filaments, and honestly, I don't think intermediate filaments are super important. I think what you need to know is that a lot of the cell's ability to resist force come from intermediate filaments. That's the one thing I remember about intermediate filaments. I'm not going to lie, everything else I don't really remember that much. But in terms of like what you should remember that in terms of the order of strength that they're able to withstand, it's going to go intermediates first, then actins, then microtubules, if I'm not mistaken. Double check me on that. I'm pretty sure if I remember right, that is the order that they tend to believe. Because, you know, the more force that you can withstand without deforming, the better. And so microtubules are terrible at that, as you can see. Microtubules take, like, no force, and they deform a crap ton before they rupture. Intermediate filaments, they can take pretty much, not infinite, I won't say infinite, they can take a huge amount of force, and they will still remain only deformed to a certain extent. It seems to asymptote around here, right? Microtubules, though, they rupture, like, immediately, and then actin takes a while to rupture, so. Someone's banging outside, I don't know what that is. Alright. So how do intermediate filaments differ from actin/tubulin? So the reason that they differ is that the monomer in this case is actually just this strip, right? And basically what you do is you take two of these alpha helical monomers and you combine them. That's pretty much it. And then what you do is with that is then you take a bunch of them and you layer them on top of each other. So there's no plus ends or minus ends with these guys. A cool thing about these is that, you know, myosin, if I'm not mistaken, the myosin heads are basically considered an intermediate filament. So, I'm pretty sure myosin, like the myosin, it's a very similar kind of process. So, yeah, but that's to say here that there's no dedicated motor protein. That's something to remember. So I know they look structurally similar to the myosin kind of tails, but don't kind of mix those two together. So the alpha helical region of the monomers of this monomer, basically, you combine two of them together, and then you slap multiple of these on top of each other, and then you take multiple of these, slap those on top of each other, you get this tetramer, and then you slap multiple of these on top of each other, and you get these massive webs of intermediate filaments. The important things to remember about intermediate filaments is there's no plus or minus ends, there's not always a head domain, so like there's not always a myosin attached, or there's not always something else attached to it. There's no dedicated motor proteins, so, you know, myosin doesn't attach to, like, it isn't just generally attached to that. Yeah, I really should specify that. Myosin is a motor protein specifically involved with thick filaments. So I keep bringing up myosin. I really shouldn't be doing that. Forget everything I said about myosin. Do not listen to that. Myosin is its own thing. It kind of looks similar structurally. That's what I was trying to make a point of, but they are not the same. Intermediate filaments are not the same thing as myosin. They're also not in the same category, period. So, back to this. Intermediate filaments are very stable. They don't degrade or get added to very much. There's no nucleotide binding domain. And the monomers are filamentous, meaning that they are strings. So this is a string, right? In the case of actin and tubulins, those are like little balls. Those are globular. So that's not the case for intermediate filaments. Intermediate filaments are these strings, while actin and tubulins are globular. So question, what makes intermediate filaments different from actin? Oh boy, there's a lot. So there's no nucleotide binding domain. Intermediate filaments don't have plus or minus ends. Intermediate filaments are filamentous, while microtubules and actin use globular proteins. There's a whole bunch of different things you could talk about there. And so here is a nice little final diagram of what we have talked about for the 3 different types of cytoskeletal proteins. And that is actually going to be the end of this video. I do hope it was helpful, much shorter than the previous ones, like half the time. But again, I hope it was helpful. If you have questions, leave comments. More than happy to try and answer, hopefully on time this time. I recognize that I answered some of the exam questions like 2 or 3 days late. Hopefully on time this time. But other than that, best of luck with studying, guys. And yeah, have a good day and happy studying.